Can we? Yeah. So first of all, thank you very much for the invitation of Fernando and Ralph. I am Leandro Barbosa, professor at the Physics Institute here at the University of São Paulo, here at the main campus in the city of São Paulo. First of all, I will introduce myself a little bit. I am a physicist. I arrived at São Paulo exactly 20 years ago, at February of 2000. I started my graduation in physics. Then I made the graduation. After that, I made the PhD, everything inside the biophysics field. During my scientific initiation, I started to work with micelles and colloidal systems and stuff like that, to see the interaction of micelles with drugs and so on and so far. And then during my PhD, I started to work with proteins as well, protein membrane interaction, protein surfactant interaction. Then in my PhD, my postdoc, I worked with membrane proteins. Then I got a position around 2011 here as a professor in the university in March of 2011. So in a couple of weeks, I'm having 9 years that I'm a professor here at the university. Every time you have some question regarding anything about this presentation, please do not hesitate in stopping me and ask. It's better because then we can discuss and talk a little bit more. So here at São Paulo, when I was hired at the university, I was hired to join the biophysics group. And the biophysics group at that point was composed by one experimental researcher, Professor Maria Teresa Lamy. She coordinated the biomembranes lab. Then we have 3 or 4 other researchers. They working with theoretical physics, statistical mechanics, dynamic simulations. And I joined the experimental group. I'm an experimental physicist, and we started to work with the biosystems lab. There in the systems, we studied several different kinds of biological relevant systems. For instance, here we are seeing, for example, an example of drug and membrane interaction, where when you use small angle X-ray scattering, which is the main technique that I have been working in the last couple of decades, that I will talk to you a little bit about this experimental technique. So using this technique, we were able to find the location of some specific drug inside the membrane. We can also study the multilamellarity of some vesicles or multilamellar vesicles. We are also, in the last few years, started to work with nanoparticles, which will be a matter of discussion for us tomorrow. Tomorrow, we'll be talking a little bit about these systems. And today, I'm talking to you a little bit about proteins. Stuff like that. For example, we have the protein A, the protein B. I'm sure that yesterday, Professor Chuck had a very, very big contribution in this protein area. So I'm sure that his classes was wonderful. They were wonderful. So he explained very well the importance of proteins and enzymes and their complexes. And we also have worked a little bit together with Professor Chuck. The main idea here is you have the protein A, the protein B, so you have the small angle scattering of protein A, the small angle scattering of protein B, and then you mixture them and try to find the AB complex to see how is it organized. Because if somehow you can stop this organization or this complex formation, you may stop, for example, the parasite to grow or to reproduce or to do something like that. Also inside this main project, we are working with a special class of proteins named chaperones. Those chaperones are very, very important to the protein homeostasis. For example, when the protein are some let's say, stressing conditions, like high temperature, changing on pH, and stuff like that, the protein overproduces some specific proteins, and those proteins, they are obliged to protect somehow other vital proteins of the proteins. Those are the chaperone proteins. And we try to find, for example, the envelope of those proteins. This also is regarding the cobblestone studies. This is trying to find or to taking a look at the unfolding process when you have the protein folded and by some reason, for example, the presence of an external agent or even increasing the temperature or something like that, the protein can start to unfold or even to aggregate. And then we are able to try to find the pathway that the protein is undertaking to go from the native state to the unfolded state. Those are some examples. You can find more stuff on the website. As I started to talking to you, I was starting this first couple of slides to introducing myself. And besides this work inside the academy, inside the university, I have also some parallel work in the Brazilian Biophysical Society. We are in the board of the Brazilian Biophysical Society for the last 5 or 6 years. And inside that society, I'm the scientific director. And one of my duties, my responsibilities, is to organize meetings and events and stuff like that. And I'd like to get this opportunity to talk to you about 2 events that we are organizing nowadays. The first one is on the right side, will take place in the end of May, from 25 to 29 of May. It's the meeting of the Brazilian Biophysical Society, but it's what we call the regional meeting of the Brazilian Biophysical Society. We have a lot of complaints from our associates that every meeting was taking place or in São Paulo or Rio de Janeiro or Minas Gerais, some of the 3 states. And because they are the richest ones, so have more research centers or something like this, so by some reason the meetings were almost all of them here in those 3 states. So they asked to us to try to get some of these events to the northeast region of Brazil, which is a very, very poor region. They have a lot of problems over there. And they were not able to get money to pay the trip, to pay the airplane, to go from there to here, because it's very far, it's very expensive. And we decided to start to organize those meetings there in the northeast region. Nowadays, we are in the 6th year of this meeting. We are making every year, we choose a different state to go with this meeting. And the main idea is to bring there nice researchers from Brazil and abroad, and the prices are extremely low to favor the participation of the local students, and of course, people from the northeast region. And we have you can take a look in this website. You can write an email directly to me. I can help you to go there, because it will be a very, very nice meeting. And the other side here, we have the annual meeting of the Brazilian Biophysical Society. At this year, we have a very, very special meeting, because we are organizing 3 meetings at the same time. The annual Brazilian Biophysical meeting, the annual Brazilian Biochemical and Molecular Biology Society meeting, and the meeting of the 20th meeting of the International Union of Pure and Applied Biophysics. This is of course, an international union held at Europe. They are responsible to try to put together the societies working with biophysics and related subjects. And this meeting takes place every 3 years. So we have now in 2020. The next one will be 2023, then 2026, and so on. And we are very proud that we are organizing this meeting. We have already confirmed, I mean today, no, we don't know the coronavirus effect yet, but today we have confirmed 3 Nobel Prizes that are coming to Brazil to talk to you. Besides then, we have very, very high nominated researchers. Please take a look there. It's a very, very interesting place, a very, very interesting meeting. And we have also some fellowships to help you student, you postdoc or you a graduate student, to go there to show us your work, to go there and see the work of other researchers. And even here, if you have some doubt or have some problem, you may write to me. I can help you with your doubts. Okay? Only to let you know, I don't know if you are from Brazil or not, these are some particular pictures of Foz do Iguaçu. We have a very, very nice bird place there. We have also our falls. I never was in Niagara Falls, but they say that it's much beautiful than the Niagara Falls, but I never was there. And we have this place for birds and several other stuff to go there. We are in the triple border of Brazil, Argentina, and Paraguay. So it's possible to take a van, a transfer, go to Argentina, take a dinner, and then come back to Brazil again to sleep. And this is very good because of the wine and the meat that they have over there. It's better than ours. And here is São Luís do Maranhão. If you are not from Brazil, perhaps you never heard about what we call Lençóis Maranhenses, are those regions here. They have a lot of sand and water, so those, let's say, small lakes, they are moving around mostly daily, on a daily basis, and it is a very, very nice place also to go there. And I think it's a very, very nice opportunity to bring our community together, in particular in this political moment that not only Brazil, but several different countries in the world are crossing nowadays. Okay? Can we start now? Okay. So, to start to talk to you about the biophysics, I think we must set a beginning point. I read a lot about this subject, and this is my personal opinion. I would say that the nanoscale, or beginning to take a look in the nanoscale for the modern point of view, for the contemporary point of view, it started in 59 with a speech of Professor Richard Feynman. If you are not a physicist, and if you have less than 30 years old, perhaps you never heard about him. He was a very, very important physicist. He was involved in the Manhattan Project. He was a very, very smart and funny guy. And in 1986, if I'm not wrong, we have a very, very tragedy with the Columbia or Discovery Airbus, I don't remember, where several astronauts died in that crash. And he was the one who discovered what was the problem. And the problem was, let's say, simple, as they have O-ring rubber. It was a very, very cold day, and that O-ring was not supposed to work in that temperature. So it was, instead of to be, let's say, soft, was very, very duct, and then it broke, and then exploded everything, and they all of that because of that. And he was the guy who discovered this after several months of investigation and so on. It's a very, very nice guy, an interesting guy as well. And he, in this speech in 1959, he said, there is plenty of room at the bottom. If you get this speech, you can find it on the internet. It's a very, very nice speech where he talks about nanotechnology, like somehow it looks like he was seeing the future. And what he's talking here, let's get a pinhead. For those in Brazil who don't speak English very well, uma cabeça de alfinete, a pinhead. We have a diameter of more or less 2 millimeters of this pinhead. If you calculate the area over there and you make some calculations, I mean, why could I don't write the entire Britannica Encyclopedia? Remember, in that period, we don't have Wikipedia yet. So we have books and those heavy stuff that we have in libraries. We have that. We use that to make research. And he say, why cannot put the entire Britannica Encyclopedia inside a pinhead? They say, come on, you're crazy. You don't have much space. Why not? It's only I use a very, very tiny letter. And if you make some calculations, you have the pinhead. You can make this calculation by your own. Those are very, very simple. You get the area, PR squared, the area of the pinhead. Then you say, let's say nowadays, you can, if you have a page book, 1,000 nanometers by 1,000 nanometers, you can read this easily. You can get any manuscript or scientific paper using cryo-EM or tomography. You can see the resolution that they have. You see, now it's okay to have this size of letter. And you may say, and you make some calculations to see the area of each page, and you see the total area, and you say, how many pages I can put here? If you do the calculation, use those values, you can put there 3.2 million pages. 3.2 million pages. Do you know the Britannica Encyclopedia, how many pages they have? Around 35,000 pages. So you can put 100 of those encyclopedias, 100 of those encyclopedias, in a pinhead if you use this size of sheet. If you'd like to go further, if you get the entire knowledge of Wikipedia, you need 2 pinheads. 2 pinheads. Suppose that everything that's there is true. Huh? So, I mean, there is a plenty of space over there, and this is what Feynman was talking about in that famous speech in 59. Why I'd like to start this conversation with you talking about Feynman? Because when you are going to study your system, it doesn't matter if you are looking from the theoretical point of view, if you are looking from the experimental point of view, you must be aware, you must know which are the important sizes of your system. You must have this knowledge to choose the proper methodology to understand each case. For example, if you have a problem or a scientific question that you are asking, you need atomic structures. I must know the position of each atom that composes my protein or that composes my enzyme or whatever you are studying. So you don't have a chance. You must go to the atomic structures. This is a quite old slide, and it's saying diffraction techniques here. And it's true. To have a to have atomic position from the experimental point of view, you must have a crystal, and you must perform a diffraction experiment, and then you can have the information of your crystal structure. However, this is the last 5 to 10 years, is it possible to use another approach, which is cryo-electron tomography. You can also get atomic resolution, nowadays around, let's say, 3 angstroms, 2.5, perhaps 2.2, depending a little bit, but you must be aware with the image computation process. So, but I mean, we can be safe on the 2.3, 2.5, and 3 angstroms of resolution. Of course, depending a lot of the size of your protein, your system, your grid, the ice quality, and so on and so forth. However, if you are looking for big sizes or larger sizes, you can also use different experimental techniques. For example, you can use, I put here SUNS and SACs, small angle neutron scattering and small angle X-ray scattering. Those are scattering techniques. I will talk a lot about them today. Actually, I will talk about small angle X-ray scattering because here in Brazil, we don't have small angle neutron scattering, at least not nowadays. In the past, I think in the 60s and 70s, they had a neutron diffractomer here at USP. Now, actually, it's in the campus of USP, but it's another institute named IPEN. It's the National Institute of Nuclear Energy Study, and they have over there, but I don't know if nowadays they are working. But we don't have this technique in Brazil. Going on, if you'd like to study larger structures, let's say virus or bacteria, and we are very, very interested in virus in the last couple of weeks, the corona effect. We can use electron microscopy, the regular one. We can use ultra small angle neutron scattering or X-ray scattering. And also we arrived at the light techniques. We must remember that we have the wavelength of the visible light from 400 to 800. Depending a little bit on your wavelength, you can be something around that order of magnitude. So, the first time, now I will talk more deeply in the experimental point of view. You have your problem. Let's say I would like to understand the structure of my protein, or I'd like to understand the structural influence of the substrate to that enzyme, or something like this. And you have several available experimental techniques. They are much more techniques than those, but this was a special issue that we published as a chapter in the book. So we decided to talk just about those techniques. You must be aware that each technique has this advantage and also those disadvantages. And you must know those advantages and also the disadvantages to be sure that that technique will fit your problem. To be sure that that technique will answer your question. And this is what we are doing here. We are making questions and trying to find those answers. For example, now I'm talking specifically from proteins. For example, crystallography. It's an amazing technique because you get the information of the atomic structure of your protein. And this is amazing because we know for a very long time, even if this is a little bit argued nowadays, we know that when we have the structure of the protein, we know its function, or when we have its function, we know that the structure is most likely in the native conformation. And nowadays we know that also the dynamics have some rules in that balance. So perhaps you have let's say 3 legs or something like this, in structure, function, and dynamics. But we have also some problems like the intrinsically distorted proteins and so on. But whatever, crystallography is an amazing technique. The only problem, you must have a crystal to make the diffraction. And to obtain a crystal of proteins is not an easy task. There is not, let's say, universal protocol to get or to produce a crystal. It's kind of try and error procedure. So sometimes you have a student taking 1 year, 2 years, 2 years in a PhD project only trying to crystallize the protein. And perhaps in the end he was not able to crystallize, or he's able to crystallize a piece of the entire protein. If he is luckily, is the piece of interest, let's say, is the piece that is interacting with some other protein or substrate or whatever, but sometimes not. So it's the main problem of that technique is that you must have the crystal. You have NMR, and it's very nice technique, and the magnetic resonance. nuclear magnetic resonance. The main is to study proteins. This is every time we can argue, we can discuss a little bit. The people that work with NMR, they don't agree very much with me. But we say that it's a very good technique, but to study what we call small proteins, smaller than 50 kilodaltons. Please, this is a general number. I'm not saying that 49 you can do NMR and 51 you cannot. This is an order of magnitude. But, I mean, for sure, 100 I never saw. Perhaps there is some, but I never saw. Yeah.
What means this units? Dalton?
Oh, kilodalton, this is a unit to weight protein. 1 dalton has the weight of 1/12 of a carbon atom, more or less a proton. So when I say that the proton has 50,000 dalton, I know that the molecular weight is 50,000 grams per mole, like the atomic unit. Okay? And so this is the problem of cryo-EM, of NMR. Then we have cryo-EM. It's a very, very amazing technique. They won the Nobel Prize like two years ago in 2017. 3 researchers because of the development, improvement of this technique. I personally quite believe that this technique will also improve a lot from the experimental point of view, the detection point of view, the electron beam production point of view. So I'm sure that will be a very, very important technique in the future. It's very nice because you can work with very, very small protein concentrations. And this is a very important point because sometimes we are not able to produce the protein at large quantities. The main problem, this is an old slide, because it's difficult to get the protein structure for smaller proteins, let's say smaller than 2,000 kilodaltons. Of course, this is not so up to date. Surely there are protein structures with masses smaller than that, but this is only to have a general idea of the technique. I will jump this one because I will come back there after. Dynamic and static light scattering, they are very nice, they are easy, they are not so expensive. The main problem is that they don't have enough resolution to get you some, let's say, useful information. You may have information regarding protein aggregation or perhaps protein dissociation, depending on the case of the protein, if I have a very huge protein, but it's not so interesting to give you detailed structural information on your problem. Then we have our technique here, small angle X-ray and neutron scattering, which is the technique that I will stay talking to you today. And the main advantages of SARCs is that you can work with several different experimental conditions, even experimental conditions near the physiological one that your protein is working on. Let's say pH variations, ionic strength variations, protein concentrations. We can work in crowded environments, and it's a very, very nice technique. The main problem is also regards the resolution that you are able to tackle. Here, using SACs, we are not able to get some very, let's say, detailed or smaller than 20 or 30 angstrom of resolution. So if you have some kind of protein structure that the main difference from the stage A to B is just a small change in a small piece of the protein, most likely you are not being able to see that change in the small angle X-ray scattering experiment. You must go to other ones. But this is what we are talking about today. This is a very common and typical SACs equipment. What is nice from this point of view is that it doesn't matter if you are inside a synchrotron or if you are in a scientific research lab. The main idea of the technique is the same. You have here an X-ray source. Here, for example, is a conventional tube, but it must be a synchrotron ring. Then you have a monochromator. When you produce X-rays, you have several wavelengths. You must select a narrow distribution, let's say one wavelength, but this is, of course, topical, but we got a narrow distribution of wavelengths. Then this guy, when goes apart from the monochromator, goes like a cone-shaped beam. We must have then a collimation system to get this beam propagated like a line or something like this to hit here the sample. So here is my sample. In this case here, the sample stayed under small pressure, let's say, vacuum or something like this, but this is a small pressure. This is to help the X-ray, because the X-ray interacts with air. So we must take out the air as much as we can to avoid this interaction with X-rays. Then we have the detector here in the end of this line. And the main difference of the different equipments around the globe, of course, relies on the quality of the materials that you produce. Your pinholes that are in the collimation system, your detector, and so on. But the main difference, in my opinion, regards the X-ray source. You may have this conventional tube, as I just mentioned to you, or you may have a synchrotron ring, as in this case here, I'm showing to you the LNLS or UV machine that we just shut down around 6 months ago, because we are building a new one. Of course, you are seeing the news. We are building Sirius. To have an idea, this is this is a reportage in the newspaper that's 3 or 4 years ago. Just to have an idea, this guy here will have around 30 meters from diameter, and here, this small spot, blue spot here, is the guy from the left slide. So this one is much bigger. We have around 150, more or less, diameter of this synchrotron. We will be able to make very, very nice experiments really in the edge of the knowledge and the science world. Here's another publicity. We see this is so the first time I went there to make some small angle measurements was in July of 2001. So we get in the last, let's say, 19, 20 years, all the changes, not only in the Sirius point of view. Sirius started, let's say, 2013, 2012, or 11, the initial discussion, something around that. Then we have the construction itself. Takes a lot of time, and nowadays we have a very, very good machine. This is not up to date, but you may see the construction a long time, and in the end of the process, we make the inaugurament of the Sirius in the last year, actually in the year before, at November more or less, during Michel Temer's government. And nowadays we are keep going with the constructions. We have the news that we have all the money, all the budget that we need to finish the construction of the beam lines the last year. So we believe that now we will be able to finish the construction of this guy here. So this is a sketch of the technique. We have an X-ray source, monochromators, collimation system, a sample, and this is the first point that we must pay attention here in the class today. Why the name of the technique is a small angle neutron scattering? Why the small angle? Because I put the detector far away from the sample holder. Let's say, again, this is an order of magnitude, 1 meter away, something around this. Could be less, could be more than that. Then I'm getting in the detector the guys that interact with my sample here and reach the detector in a very specific position. We are looking for 2 theta on the order of magnitude of up to, this is our maximum, 5 degrees. So we are working from 0 or something really close to that up to 5 degrees. This is why we call small angle neutron scattering. And when you are looking at that region, that angular region, you are able to get information about, let's say, strokes from 20 to 500 angstroms or 2 to 50 nanometers. This is more or less our window of view. If you have some particle or some information that do not realize in this interval, you perhaps small angle sketch is not the most suitable technique for you to use. But generally, we stay more or less in this regime. So the main idea, we have the X-ray beam. We hit the sample. We have the sample and my scattering particles over there. Here, the buffer or the water solution or whatever you have is represented by this blue color. The red particles are my scattering particles. I make the experiment of this system. Then I make the measurement of just my buffer solution or buffer or water system or whatever. Then I subtract this contribution from that contribution. And then I have the contribution of my scattering particles. This is in general words, more or less what we have here. For example, here is the scattering pattern of BSA, bovine serum albumin, a very, very well-known protein, a module protein for several experimental techniques. And here we have the buffer contribution. When we subtract one to the other, we have the scattering of the protein here expressed in those blue dots. When we have this guy, then we start our data analysis, and this is what we are going to do right now. Okay? I don't want you to write in those formulas. This is not, of course those are important, but this is not important for the scope of our class today. You can write an email to me, I can send you the presentation. There are much more formulas if you like formulas. We don't like you to be afraid or something like this. But what we have generally, this is the main equation of small angle scattering technique. What I'm saying here is that my intensity is something, is proportional or it's equal to the Fourier transform of my electron density fluctuation. This is, you can find this in several review articles, in several small angle scattering books. You have the first book of small angle scattering from 1955 from Professor André Guinier. It's an amazing book because despite the notation that is not so easy to understand, when you understand well, or understand quite well the notation, then the book is really, really good. He's very, very nice. After that, in 82, we have the book of Professor Glatter and Craddock and Glatter, and then in 87, the book from Professor Svergunens. You have several books after that. Those, you have review articles. You don't need to worry about that. What I would like you understand today, we have our scattering particle. We have the idea that I have a scattering particle inside a continuous matrix. Let's say a water matrix, and inside that a protein. Could be whatever scattering particle you'd like to, but we are treating here scattering particles inside a matrix. When I have this, and now it's the sphere that we physicists and our engineer we like very much, I will say, or I am inside the particle, or I am outside the particle. I have an abrupt discontinuation between the surface of the scattering particle and outside. This guy here, those fluctuation, is the fluctuation of the electron density of my scattering particle as compared to my buffer solution, my buffer contribution. This arises from the fact that X-rays, they interact with electrons. If the guy has electrons, we will be able to interact with X-rays proportionally to the number of electrons that the guy has squared. This is more or less what the quantum mechanics talks to us about this interaction. So what I'm seeing here is the electron fluctuation that those guys have over the buffer contribution. This I would like you to know. So you must know by now that the contribution of the scattering from the electrons that compose the buffer, I have only the contributions of the electrons, those over contributions, those fluctuations that I have of the scattering particle as compared to my matrix solution. Why this? Because this is important to split some contributions. Now we are making an assumption. We are making that every time an electron, a photon, reaches the detector in a specific position, I have an interference problem, and in those interferences, those will arise from two different electrons inside my scattering, inside my system. I have now two possibilities, or this pair of electrons, they are inside the same scattering particle, or they are from two different scattering particles. We try to deal with those stuff. Then we have two main contributions. Those electrons that are inside the same scattering particle, those give rise to what we call form factor. That information will give to me how is those electrons spread around inside the scattering particle. Of course, we have a lot of approximations inside the theory. A monodispersed system, every scattering particle is the same, and so on and so forth. And when we get information of two different electrons, we are talking about the structure factor. Now I get information, how are those scattering particles distributed as compared to one reference scattering particle. We are not talking about that today, a little bit tomorrow perhaps, but today we will focus our attention in the form factor problem, which means I have a scattering particle, I don't know a priori the shape of the scattering particle, and I must discover which is the shape of the scattering particle. This is what we are doing today. Okay? So again, just to put some formula for you, but this is what I would like you to call your attention, is the scattering intensity is proportional to the density, the numeric density of the scattering particles, which means the number of the particles that are being illuminated by my X-ray beam. It will be proportional also. It's missing in square here. The electron density contrast, which is the difference from the scattering particle in the matrix, the scattering volume, and the form factor. For systems that are very, very diluted, which means that the particles are very, very far away one from each other, it's possible to say that T sub Q is equal to 1 because of the G of our function. We don't have to worry about that now. Just believe me that this function is 1, and we are not worried about it today. So what we are looking for is the scattering amplitude, which is the electron density difference between the scattering part and the solvent, the Fourier transform of that function over there. When we don't have, so that is all the theory that you must know to understand the other part of the classes. Of course, we can talk in the afternoon. I'll be around to the end of the day today, and even tomorrow, even Friday if you need something. So don't worry. I would like to say, okay, professor, you do that, but I mean, how can I use that information to understand my problem? I mean, which is my problem right now? I would like to know which is the shape of my scattering particle, dot. Let's say I make an experiment. This is one possible treatment. I make my experiment. I say, okay, which is the formula? With physicists, every time we like to start with spheres. Let's suppose we have a sphere in solution. This would be like a hard sphere in solution, a snooker ball in solution. I have the electron density here. It's equal to a constant value if I am inside the scattering particle. It's equal to the water if I am outside the scattering particle. A very, very simple step function, of course, spherically symmetric. So I know that my intensity is proportional to the Fourier transforms, the last slide that I showed to you, of the electron density of the scattering particle minus the electron density of the bulk contribution. Okay, then I have to put, I can substitute those values. If I have a sphere, and this is the very first reason that we like very much spheres, because we have a triple integral to make it here. We have the R contribution, the phi contribution, the theta contribution in the integration, but if you have spherical symmetry, if you have a sphere, those contributions go away and they became a 4 pi times R squared dr. Then we have also the information that when we have a spherically symmetric Fourier transform, it's possible to get the expansion of that guy here and put the sin, the QR divided by QR. This is a very good information for this stuff. And this is an integral, despite it's quite ugly. It's not so difficult to solve. You can use what we call parts resolution, 2 times. It's 2 times because of those power here. But I will not be very much on that. Don't worry about this. You must just would like to reach the final result. It's not that difficult. Just to make some math, it's not that difficult. When you make all of that calculation, you reach something similar to that stuff here. You have 2 times the volume of your sphere times the electron density contrast. Then you have the multiplication of, this is a very, very important equation in scattering experiments and also in EDPs, because it's a solution in the Bessel-like way, like the Bessel functions. You have the sine of QR minus QR times cosine of QR divided by QR at the 3rd power. All of those stuff here is squared. This is the scattering of a sphere which radius r in solution. If you get that, you have that information. Of course, you will remember your course in your faculty or in your school. We have here sine and cosine. Those are oscillating functions. Since they have this kind of information, perhaps they will have some values that this guy here will be equal to this guy here. And when you have this subtraction, you have this is equal to 0. But since I have the squared here, everything is positive. So this stuff may oscillate. And if I put in the log scale in the y direction, it's nice because when I have 0, I don't have the definition of log of 0, so I have a minus infinite function. So I may see some oscillations in our scattering curve. Let's take a look on that. Here is the red line is the theoretical calculations. The experimental dots are the blue and the green one. The blue is Silic spheres. They are very, very nicely produced to be very, very monodisperse. I mean, all of them, they have a very, very small radius distribution. And this is the theoretical approximation that we have. As we expected, we have some minimums here, that those are reaching minus infinity, because we have a log scalar here, and we have a function that have two oscillation functions, one minus the other. Perhaps we may have some values that one is equal to the other, and then we have a 0 here, and we have this kind of effect. So we have also, so we can see that this approximation is very, very good to treat spheres in solution. But of course, our world, unfortunately, there is not so many as examples of spheres in solution. And we know that this model is not enough to describe more complicated or more interesting systems. But we like spheres because then we can go to the other one, which is the ellipsoid. So perhaps you may not have an ellipsoid, but you may not have a sphere, but perhaps you may have an ellipsoid. And this is quite interesting. I will not again show you the equations. I don't care about them. You can find those in the book. There are some theses of some theoretical physics that the guy do everything step by step. You don't need to worry about that. It's important to you to know that those stuff are solved somewhere. If you'd like to do it, it's nice because then you can find the proper solution and compare your result with the result of the other guy. But the main idea here is that I have a sphere with equal cross-section. This is our arm, and this is a slightly, let's say, bigger or smaller, but I mean, this is a constant times arm. If this constant is bigger, larger than 1, we have an prolate object. If it's smaller than 1, we have an oblate object. Just you to remember, this is an oblate object. This is a prolate one. Okay? So, you do everything again. This is called the geometrical procedure to treat the small angle scattering. You define the geometry of your scattering particle. You define how the electron density of your scattering particle is spread all over your system. Then you start to make those calculations like a crazy person. And then you can see that you have several different stuff. Here I put only the final solution. When you have all of those steps, this is not so easy as the first case. You have to take some other stuff in consideration here. Mainly is that when I have a sphere solution, doesn't matter the relative position of the sphere as compared to my incident beam. Now, since I have an ellipsoid, it will matter, because you must remember we are seeing the cross-section of the scattering particle. If the ellipsoid is in that position, or in that position, or in that position, or in that position, I may have different contributions. So we have to make this what we call ensemble. A sum of this ensemble of different possible positions. But it's possible to treat analytically this equation. And then you can try to say, okay, now let's go to a more biophysics example. I have here the scattering solution of a very, very well-known protein named lysozyme. And this protein has a quite globular conformation. Here are the experimental dots. And this black solid line here represents the represents the small angle scattering of some ellipsoid with R equal to 15.48 angstroms, and an isotropy, which is that value that I mentioned to you, 1.61. So I'm not what I am able to say is that lysozyme in solution behaves just like, from the small angular scattering point of view, just like a bench of ellipsoid with those characteristics over there. This is the way to treat your scattering data using those information. For example, perhaps for you, that information is enough. Now, if I know that the protein that are produced is in the monomeric form, for me it's fine, I don't need anything else, I can move on to other relevant scientific question. Or perhaps I say, no, this is not enough for me. I must have more detailed information regarding the structure of lysozyme, so I must go deeper in my analysis. I must try to get some more information of that scattering curve. Okay? So, concerning the scattering curves of protein in solution, I was talking to you about this first way of analyzing the scattering data is what we call the geometrical approximation or geometrical model, where I define the model of my protein and then start to make that calculations and try to compare the theoretical, the possible theoretical conformation or geometric object with my scattering particle. When you are in the real world working with proteins, this is not so easy. Generally, they don't behave like lysozyme. Lysozyme is a small, very, very, very well-behaved protein. Generally, the world is not simple as that when you are studying proteins in solution. One thing that's really, really important and takes place in the last few years was to study the folding problem or the unfolding problem or the problem of the intrinsically disordered proteins. This matter a little bit of talking between us because we discovered in the last decades that there are some specific proteins that are related to some specific diseases, and with special attention today from my presentation to you about the neurodegenerative diseases like Parkinson's disease, Alzheimer's disease, Huntington, even some types of diabetes and so on and so forth. Depending on the protein involved in the process, you may have one disease or another disease. What is interesting is that the general, please, I'm talking about like a physicist now, huh? Not a biologist or biochemist. The general point of view, the behavior of the disease is very, very alike, it's very, very similar. You have a protein or a bunch of proteins by some reason, don't know, we know some reason, but I mean, this is not very, very clear for us today, not yet. We have some structural changes from these proteins, and they start to aggregate in a very, 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 very specific way. And this we can say, doesn't matter if you're talking about the Parkinson or Alzheimer or Huntington or diabetes, the structure of those structures, there's what we call cross-beta structure, they are alike. They are the same from the sub-Youngstown position. They are the same. So this is why, as a physicist, we talk of this, let's say, general structure, but we know that each disease has its peculiarity. So what we'd like to know is how you can go from the protein's okay, it's happy, it's work, and fine, then by some reason changes its structure, or entirely or locally, then you have the beginning of an aggregation process, and then you give rise to the disease. And this is the same mechanism for Parkinson, Alzheimer, all of them. The differences are regarding the protein in the process, in the main protein involved in the process, the other proteins involved in the process. But even when we discover that stuff, increased a lot the interest in trying to understand how behaves the unfolding proteins in solution. How are they behaving from the scattering point of view in solution? So we have here the Craddock representation. This is very funny because Professor Craddock, a very, very good researcher from Austria, he is from the, let's say, 40s, 50s, 60s, something around this. He was a researcher working with polymers in solution. He made several seminal contributions in the polymer area of small angular scattering. And in the beginning of the 2000s, or even a little bit before, people realized, physicists realized, because we'd like to try to make easier our life, and I'm very happy that Chuck is not here today, that proteins are just like biological polymers. They are not. We know that they are not. But why I'm saying this? Perhaps I can get the information of Professor Craddock regarding polymers in solution and try to apply two proteins or unfolded proteins in solution. And this is what we are doing here. We have the Craddock plot. It's just a different way to represent your scattering data. It's very nice because if you do a plot of Q square times your scattering intensity, those guys is very similar to this here, but this is the, let's say, the regular or linear representation. Here I have the Craddock representation. So Q squared times the intensity against the scattering vector. Just taking a look of your scattering curve, we may be able to see some information. For example, if you have a kind of a plateau around here, you have an unfolded protein behaving just like a polymer in solution. On the other hand, if you had something similar to a Gaussian-like or a very symmetrical peak, you have a globular conformation, which from the Professor Craddock point of view was what we call a polymer in a bad solvent. Because of the polymer science, they have a very nice nomenclature regarding the structure of the polymer. If you put the polymer in solution and the polymer is completely unfolded, he's in a good solvent because he likes to be there. If you put that in the polymer does like, it remains like a small sphere, he's in a bad solvent because he doesn't like to be there. So we get that idea to talk about folded protein, unfolded protein, and this is the main point of our research nowadays. One of the main points is the partially unfolded proteins. Because the protein is not completely unfolded, but is not completely globular. Could be a protein, for example, that you have two globular domains attached with a disordered linker or similar to that. So this is a very, very nice information that we may see some examples after all. Another, you realize it because when we talk about the Craddock representation, you don't have to do any calculation. You must have to represent your scattering data in a different way. Instead of put intensity against the Q vector, you put the Q squared times the intensity against the Q vector. It's just a different way to represent your scattering data. You are not making any calculation over there. We have another structural information regarding small angular scattering. This was the first contribution of Professor. He made so many contributions. We don't know. I'm not sure if he's the first or not, but a contribution of Professor André Guinier. He was the first one to theoretically derive all the equations regarding scattering experiments. Then we have this duration radius. This is some concept that we get from classical mechanics when you are working with inertia, the moment of inertia, and so on. You have the radiations, the radius of duration. Then we get the same information from here because he realized that when you have your scattering intensity, is it possible to make some assumptions? If you take a look in the very, very small Q regions, which mean that we are near zero, which mean we are near our direct beam, that small piece over there, is it possible to approximate with a Gaussian-like function? And in the argument of this Gaussian, you have the radius of duration. So making a simple experiment, we have this is the Guinier law. If we get the natural logarithmic in the both sides of this equation, we have a straight line. Now, every time you see some equation, you see some theorems, whatever, you must try to put yourself in the historical point of view. What were those guys and girls living in that moment? Why he was looking for something to linearize? Because equations we know how to treat. We know how to fit the best equation to get the linear and angular coefficients. Nowadays, of course, it's much easier. You get a computer. Perhaps a cell phone is able to make a fit. But in the 30s, 40s, 50s, it was not that easy. In some places, you don't even have calculator machines, so you must have everything in your hands. So they were trying to find stuff to linearize everything. So this is why in the beginning of this century, in the first decades, they try to do this kind of approximations to try to get more information of their scientific systems. And everything of that is true if you are inside that approximation over there, which is that this is true up to that limit of Q times the radius of duration smaller than 1.3. This is also, again, something around that, but you may not go further larger than this because you will be away from the Guinier approximation. if you realize in the last slide, I showed you not only the Guinea approximation, I showed you the Guinea approximation, but not only the radius of duration is a quantity that you may have, you can calculate from the scattering curve. It's also possible to get the further scattering intensity, which is the intensity at zero angle. And why this is important, because from the protein point of view, this number is related to the protein molecular weight and also to the protein concentration. So getting that information, it's possible to calculate the protein molecular weight, which is really, really good for us, because when you have a protein aggregation, it's not so difficult to realize. Light scattering experiments may be enough you to realize. The problem is when you have a partial oligomerization. For example, if you have a protein that is a homotetramer in solution, but for some reason you are producing, or I don't know why, you have the coexistence of dimers and tetramers. There are not so many experimental techniques able to say the difference among them. And small angle scattering is one of them, because you can have the calculation, you can theoretically calculate, for example, if you have the information, the scattering intensity of the dimer, the scattering intensity of the tetramer, perhaps even make a composition and say, oh, in my system I have a 70%, as an example, of dimers and 30% of tetramers, for example, or monomers, I don't know. So SACS is an important technique to do that kind of oligomer equilibrium. This is not aggregation properly, but it's a kind of a equilibrium. And this is also an interesting information that we are able to take out. This I already, just to show you an example, this is a scattering curve of a very, very, very well-behaved protein. This is, we are talking about that protein in a while, but it doesn't matter which is the protein, what the protein does. Which matters that when you have the natural logarithmic of our intensity against Q square, we have that straight line here, you see? That straight line is the Guinea law. Then we make that linear equation, linear fit over here, then we get the information of the forward scattering intensity, and you get the information of the radius of the duration. Okay, it's fine? Just to see, this is the radius of duration, something around 26 angstroms. So, after the radius of duration, something else that we have in our scattering intensity is the pair distance distribution function. Again, don't worry about the equations and this kind of stuff. Just the physical concept is important for me, that you understand. The pair distance distribution function is proportional to the probability of finding two pairs of scattering centers inside my scattering particle. To calculate that probability, you have the same scattering particle. You fix one and you start to shift the other by, let's say, R, the center to the center. Then you calculate which is the common volume of them. That volume is directly proportional to the probability of find two pairs distant one from each other R. Then you are increasing that distance. Why this is important? Because this is related to this gamma function, which is the direct probability, or most similar to the direct probability. But the P of R, this pair distance distribution function, is the Fourier transform. You see that we are talking a lot Fourier. We are talking a lot about Fourier here in small angle scattering experiments, or even in the diffraction experiments. This is why. This is because the measurement that we are making, we are not making them in the direct space. We are in the reciprocal space. Our unit of length is Q, is not meters. They are attached one to each other by this Fourier transformation. This is why every time you are seeing Fourier transform to go there, or anti-Fourier transform to come back, stuff like that. Here, for example, R. When you see R, you are in the direct space. When you see Q, you are in the reciprocal space, and you can go from one to each other, making these calculations. Of course, there is a lot of theory behind that. This is just to explain, waving the hands to you. But the most important thing is that with the P of R, if I can make the calculation, and pay attention that this guy here is an experimental information. I make the experiment, I have that information, I have the intensity. Now, if I put the intensity inside of this integral, this machine, and calculate all of that, I may have my P of R. So, the P of R is related when you fix a specific size, let's say 50 angstroms. How many times or how many pairs I can find of 50 angstroms inside my scattering particle? Of course, I can do this until I reach 50, then 51, 52, 60, 70, and so on, until I will reach a maximum value. After that value, I will be one time inside my scattering particle and the other time outside my scattering particle. I don't want to be outside my scattering particle because I don't have that information. Then it's also possible using this procedure to calculate the maximum dimension of my scattering particle, or the max. This is a very nice figure. This is the gamma of R. For example, when gamma 0 of R is when 0 is equal to 0. I have the 100 probability. The two shapes are one over the other. Then I started to split them. I calculate the common volume. I put here inside this histogram. Then I go there until I have some point here after 2 radius. The first one here and the other one here. 2R, or the diameter. I am outside the scattering particle, so the probability of finding 2 elements inside the same scattering particle vanishes, goes to 0. So this is the gamma of R, and the P of R is the gamma of R times R squared, just to make our life better, because I have here I didn't put here, here. I have here R2 gamma of R. I call it this of P of R, just to make my life easier. And I have this is why this guy goes like that, and this guy goes like that. It's just get this one here, multiply by R squared, you have the blue scattering curve. Okay, Professor, why are you almost half an hour talking about this boring function? Because when you have the P of R, you may have some interesting information regarding your scattering particle. This is a very, very nice slide from Professor Zvergun. For example, first of all, the color here is the same shape as the color here and the color here. Here I have the geometrical model, here I have the scattering curve, and here I have the P of R. For instance, if you have a sphere, all of them have the same maximum dimension, the scattering particles. If I have a sphere and I calculate theoretically the experimental scattering of those spheres, I have this black scattering curve over there. I can say again, I have the several minimums. We already saw that equation. I have the sines and cosines. It's fine. You have the minimums over there and so on. However, if you have stuff like this, 2 small spheres attached to each other, this could be, for example, a protein dimer. Why not? And you calculate the scattering intensity. You see that it's very, very similar to the scattering intensity of the sphere. It's not possible to take a look in those scattering curves to say which is the best geometry that fits my scattering particle. So, because we are in the reciprocal world, what do? Let's go to the real world, the direct one. We calculate the P of R of those structures. Pay attention in the P of R of the sphere. It's a very, very similar to a Gaussian-like function. It's not a Gaussian, but it's similar to a Gaussian function. It is very, very symmetrical. And take a look in those dumbbell structures. We have a first peak in a very, very smaller position as compared to the sphere. Why a smaller position? Because this guy here is seeing 2 times smaller spheres. Then we have the contribution of the different spheres, which gives rise to another bump over there. So when you take a look on them, you say, oh, sure, in this case I have a similar, let's say, dumbbell structure. In the other case, I have a more globular protein conformation. Of course, those are just general examples. If you have, for example, a cylinder, you have a peak shifted to smaller values. Those represent more or less the inflection point over here, the cross-section of the cylinder, and the length of the cylinder is represented by the max. Also, actually, it's not the max, it's the max, but taken in, it's the main diagonal of the cylinder is the maximum value for this scattering particle. Okay. So why I'm talking everything I'm talking everything for you now. It's because we are now taking a look at some examples from the real research, and we are trying to use those stuff over here. The first one, the first one regards scientific collaboration with us and Professor Júlio César Borges from the Institute of Chemistry in University of São Paulo as well, but in the campus, in the country part of the state, named São Carlos. He's working with those chaperone proteins as well, and also proteins related to those chaperones. And he coordinates a biochemistry lab, a protein characterization lab, and what they do, they are able to induce the protein production inside, let's say, bacteria or something similar. Then he's able to get that bunch of bacteria that was produced in his protein. He's able to purify and get only his protein inside that bunch of stuff that is over there, and then he starts to characterize not just the function of the protein as the structure of the protein. And this is the time when he called me and said, oh, can you help me making some small angle scattering experiments? I say, oh, yeah, why not? Then we get the protein, we go to the synchrotron and make the measurement. This is the scattering curve of this protein. This is from Leishmania brasiliensis, and he comes from this HSC70 interacting protein. So this is a protein that is interacting with HSC70, which is a very, very important chaperone-like, and we'd like to see how is the structure of this protein to try to avoid them to interact with the HSC70 chaperone. So this is from the scattering curve. This is what we call the P of R calculation. We get the experimental scattering dots. We make the Fourier transformation. We calculate the P of R. We have to make some trials and so on. This is fine. It's part of the job. Then we get something around here. And you look and say, okay, Professor, this is not even similar to what you saw to us just now. Yeah, it's not, because this is a real example. There was working with spheres and dumbbells and cylinders. Here we have a different stuff. But what do we see here? I have a maximum dimension around 200 angstroms or 20 nanometers. I have a maximum frequency around, let's say, something around 50, 60, 70, something in that region. This means that I have some elongated structure, not properly a cylinder, but something very, very elongated. And as I mentioned to you, you have the geometric model composed by small dummy spheres or whatever. Then you are able to calculate the P of R. It's also possible to do the other way around, having the P of R and calculate the ensemble of possible spheres at specific positions that could represent that scattering curve. This is called the indirect Fourier transform procedure. Here, for example, is what we did with that protein. These guys here, all 6 figures are the same. They are just turned one with respect to each other. And that average structure here, if you calculate theoretically, the scatter will have something similar to that black line over there. We also use the radius of duration to see the unfolding of the protein. This is used in circular dichroism. This is used in fluorescence. We can see that we have, depending on the external agent, using urea or even guanidine, we may have 2 different transitions. And then using small angle X-ray scattering and also using urea, we were able just to calculate the radius of duration of the protein, just in using the radius of duration. Here is the Guinea plots. We plot the radius of duration against the urea concentration. We can see, we can observe, let's say, a sigmoidal-like structure. And we see that the point that we have, the inflection here, around 2.6 or 2.8 moles of urea. This is the concentration when I have 50% of my protein unfolded and 50% folded, like a 2-state model. It's very, very similar of what they observe using fluorescence and CT. So somehow, and this is our case in that example, we were just interested in get the protein envelope, which were those yellow dots, and also the protein response as compared to the urea and guanidine. We are not able to measure the guanidine unfolding because the energy of the X-ray that we use here in Brazil is on the border of the absorption of the chlorine ion. So as the guanidine has a lot of chloride ions, we were not able to make the measurements. And this was the first one to show to you how you can, just by using the radius of gyration, get the information that you need depending on the answer that you must, depending on the question that you must answer. Now is the construction of another enzyme, Tcs Trypanosoma Cruzi. This is an enzyme related to the Trypanosoma Cruzi. Also, again, a collaboration with Julio Borges. So we went there, first time we got 3 different protein concentrations, make the experiments. We were in some specific condition because we were studying a different giant protein in that period. And we have that scattering curve. First of all, as you may see, you have some deviation of the scattering points when you have the intensity divided by each protein concentration. This goes up to 74. This is an indication, this is a fingerprint that I have a protein aggregation. So we are working with the system at 5 milligrams per ml, where we don't have the aggregation, you have a nice behavior. The main problem is that, you remember, I talked to you, we see oscillations in the scattering curve. We are not seeing any here. Why? Because we were making an experiment, not in a very specific conditions, because we were studying a different protein, and I have some information from that, some scattering that was going far away from the detector. Some information regarding the structure of my protein. So in order to get that information, what we should do, we should approximate the detector, change the experimental setup, to be able to get information in that order of magnitude. This is why you must have some information a priori from your experiment. We know that we were not in the very good case because we were studying another different protein. We just would like to make some tests to see if we have the good concentrations and if you had protein aggregation. When you go to the correct position, look at that. This is the same scattering curve of if you realize here is 0.15 and 0.15 is something around here. You see, here, more or less. All of that information I was throwing away because I was not in using the correct sample to detector distance. I was not in using the correct setup experimental conditions. Now, when we have a very, very good scattering curve, just like that, this is a very good scattering curve, we start our procedure, and here you may see we have a maximum dimension around 70, 80 nanometers, a main position around 30 and something. I have a more globular-like structure, but it's slightly elongated, but more globular for sure than the last case. And we get, and you say, okay, but with that, how can we be able to get the ensemble, the conformation, the protein envelope? Why we were doing everything regarding this protein? Because it was a very, very important protein on the cycle of the Trypanosoma Cruzi, and it does not have the crystallographic structure obtained yet. So we were hunting a lot to get this information and try to put in the literature. Then we start, now I will explain to you more or less how it works, the process that get a scattering curve, get the P of R, and using the P of R, try to get the ensemble of possible positions of different spheres to try to reproduce the protein envelope. We started with a very, very general and spherical symmetry with different positions. What the program does, it takes out some of these balls, makes the theoretical calculation of the scattering curve, and compare with your experimental data. Oh, it's not good. Okay, so let me take another part here. I do everything again, make the calculation, compare to the experimental data. Oh, it's better. Then I keep this one, throw the previous away, and try to move in another different part of the position. And you are doing this recursively until you reach a final position and say, hey, take a look, that position here fits exactly, it fits quite nicely your experimental data. As you may wonder, we are talking about hundreds of different positions, of different spheres. Of course, we don't have a unique solution. We have to reproduce this process a lot and try to get an average. Every time I reproduce the process. I don't have the exact same solution. You see, here I have this these red points. Here I have the green points. But if you realize, from the general point of view, they are very, very similar. I have a larger structure here. I have some stuff over there. They are similar to that. And this is the information that I can reach using a small angle schedule. I'm not able to get some more information than this. This is 2 times. We reproduce this process 40, 50, 60 times. I put 4 here to you. Again, the red one, the green one, the blue one, and the pink one. Again, none it's equal to the other one. They are all different among them. However, if you take a look in the general position, you have something which is globular over here, and let's say there is some small piece or something around this. And this is a quite a funny process. We have the software. When you start, the software just started to run, then it starts to move these small spheres to different points and calculate every time the new theoretical P of R and calculate and calculate and calculate and calculate until you reach that position. Then the program stops and say, okay, this is the best I can do. I cannot do better. Then you get the solution, you save that solution, you come back to the first point and you do everything again. Since you are using random numbers to begin this first movement, you'll never get the same position. It's very, very unlikely you'll get the same position. And then we were very happy because, coming back to our scientific problem, we were able to say that this protein has something, let's say, globular over here, some stuff over there. We at that point knew that the protein function was relating to a zinc finger. So in the absence of zinc, you have kind of a finger open like that, like a hand conformation. And have here the hand and here the zinc region. In the presence of zinc, do something similar to this. We were writing the manuscript at that period, and at that point we discovered that a couple of weeks before, a Japanese group, they were able to crystallize the protein, and now we have the protein crystal structure. So it's kind of boring for us because they have more precision than we have. And this is, just to make it a comparison, this is one of the structures that we generated. This is the protein structure that they propose. And you see, yeah, but they are not seeing that region here. Yeah, it's because to do the protein crystallization, they used zinc. So they have the zinc finger position in the closed conformation. Then we change a little bit the information of the manuscript. We change a little bit, let's say, the shape of the manuscript, make several other experiments, and compare a little bit the influence of substrates in the protein structure. This is just to mention to you that it's important to have a very, very good knowledge also of the literature to try to avoid some of these problems regarding the publication of your data. Fernando, até uma, né, que eu falo. Now, we are still in this Chaperona work. So this is from you that are from Brazil. We have, from the last 5-6 years, a research project with Professor Carlos Ramos from the Chemistry Institute of Unicamp, which is what we call a thematic project from FAPESP, where we are studying this chaperone-like proteins. Among those proteins, one that's very, very crucial, has a key role in several different processes of the cell homeostasis and cell multiplication and so on, is the HSP90, which is the heat shock protein 90. This protein is an ATPase, which means that it has a specific conformation, but in order to do its activity, it must use the cleavage of an ATP in ADP plus phosphate. And in this work, we are trying to understand, and this is the protein, very important and so on. We have, it's actually quite old publication of Carlos and Julio. This is at that period, 8 years ago. Here in the center, you are not seeing, but it's not important you to see, is the HSP90. All of those spheres, circles that you are seeing here is one different protein that is known to interact with HSP90. So it's a very what we call promiscuous protein because it interacts with several different proteins, and this is not very common in nature because when you lose your specific, you may have some, you would like that the protein do some kind of function, and at the end it does another different one. But it's not important for us. We have several different proteins that are target or client proteins of the protein. And each different colors that we have here are different systems that we have regarding the protein. For example, this yellow here regarding tumor promoting inflammatory, this enabling replicative immortality here, and so on and so forth. So these proteins are very important one and understand its behavior, or even who it's interacting with the protein. It's very, very important for us. Here in this work, we studied the activator of HSP90 ATPase1, or AHA1. This protein, what we were studying is by some reason, when we have the production of this protein, we start the ATPase cycle of HSP90. So we would like to know what is the protein doing to HSP, where is the protein attached to HSP, and what it could be done to or doing to HSP in order to activate the protein. And remember, activating the protein, you have all of those stuff that can happen inside the cell. So if you have to activate the protein, you are having some problem inside the cell. The main idea of this study was then to get the information regarding from Leishmania brasiliensis again of HSP90. It's important that this is a very, very old and well-conserved protein inside the cell, so you can find in every mammalian cell, bacterial cell, plant cell, and so on. So it's the HSP90. So it's a very, very old, from the evolutionary point of view, protein. This is what we knew at the time we started those experiments. We have this activated HSP proteins or AHA proteins from different animals. We have from the Leishmania, the sequence, from Leishmania brasiliensis, the human, yeast. We have so this kind of information. This is the work that Julio and Carlos, they are performing in their labs. They get those proteins, they find in the genome, they try to express, purify, try to compare if we have analogous proteins in systems, in the system. And what they found is that the N-terminal sequence of the LB AHA1 was like 35-40% similar to the yeast N-terminal region, to the human N-terminal region. And they found that the C-terminal region was very, very similar to the yeast one. As I started this speech to you today, I was talking about the paradigm of the function and structure relationship. What do we know nowadays? If you have the protein sequence, if it folds in a specific conformation, most unlikely, it's very, very unlikely, if you change one or two residues, you change the protein structure. Most unlikely, we have some very, very, for example, anemia falciforme, do you know the disease? You have just one mutation in hemoglobin. So it's very, very important, the mutations. But most generally, when we have the primary sequence of the protein and its primary sequence is similar to other, most likely they have very, very similar structures. So we are proposing that we have the N-terminal structure similar to the human, the C-terminal similar to the yeast, and from that in mind, let's start to make our calculations. Oh, and then we realized, we knew in that period that we have a random conformation in the middle of these two domains, like a linker with no specific or random structure in solution. We have this scattering curve every time following the same steps. We have this scattering curve. This is similar to the P of R. Here the P of R is most ugly, something around 140, maximum dimension around 30. You see that I have a very, very elongated structure because the cross-section, which is something around here, 40, 35, 30 something, 40. It's much smaller than the maximum length of the protein. We try to do, again, the problem with the small balls, and we get something similar to that. A height elongated protein, but we must remember that we have a linker position in the middle of the protein. When we saw that, we tried to use a different approach that was developed earlier, a couple of years before we started this work, which was the ensemble optimization method. This was developed by Professor Svergum team. He has a very, very good research group regarding small angle scattering. If you have some time or if you need, you can write to me. I can send you the link to his website. This system is specific when you have the crystallographic structure of different domains of the protein, but it's a very, very flexible protein because it's attached with a linker. It looks like very, very much our system. What it say? Okay, you have any possible configuration of those guys, any possible configuration. Actually, some of them are forbidden due to their Hammarskjöld plot, but we are not going into details. But I will generate a pool of 10,000, 15,000 different structures, and then we are putting them together, mixing them a little bit, and try to find a pool that represents your scattering curve. So, for example, this pool here, and all of those structures are statistically equally probable, okay? Let's say it's something that I have a pool, and I have all of those structures over there. So they both N and C terminal could be very, very far away, or even very, very attached one to each other. And when the guy calculates the scattering curve of this pool, you have something like this. Again, this is not an unequivocal problem. It's not, we may not have just one solution. It's very, very likely that we have several different solutions giving you the same scattering curve. But when we tried a little bit, when we have similar structures, we realize that the small hat that we have over our structure, most likely is that this structure is doing something like this in solution. So SX, small angle scatter, is an average technique. I mean, the X-ray is illuminating all of those structures. One is like that, one's like that, one's like that, one's like that. For me, from the SACS point of view, of course, it's something similar that are kind of dynamics, because all of those structures are equally probable. So in this study, we were able to find this kind of information. I will talk to you about the last two examples, then we close the day, okay? Yeah? The last one is just to show to you that sometimes life can be tough in research, in science, because we sometimes may have very, very difficult problems. Just sometimes, Barbara, just sometimes. Here, for example, in this work, this is not a work from myself. This is from a research group from Italy that I work together them, and this is a very, very beautiful work. I like very much to show this. This is a protein. Every different color you are seeing is a different monomer of the protein. So it's a very, very huge protein with a lot of monomers and so on. They studied the influence of pH and temperature of this protein. I will just show you one figure of this protein. Is this. When they make the experimental curves changing, for example, the presence of the buffer and also the presence of counterions, as we saw in the previous presentation, the presence of different counterions, of course, may change a lot your system, and here we have the same stuff, your biological system, your scattering curve as well. And here, the guy who make the analysis of this data, he realized that he may have 7 different, he called monomeric contributions. And 1, 2, 3, 4, 5, and 5 different of those other different contributions, because he was elucidating a dissociation process. Using this procedure, he was able to say that in the presence of this guy, for example, everyone is in the position here, representing the monomer, for example. Here, for example, we have a mixture of native-like, something in the middle here, and also the monomer contribution, and so on and so forth. So using this approach, which is QuaFit, the software that he developed, it's possible to make the composition of different scattering curves when you know the crystallographic structure of them. So it's not that when you have the crystallographic information, you cannot apply small angle scattering any longer. This is not true, depending again on the answer the question that you are answering in that specific case. The last example, this is just one slide, is regarding something that's very, very important for us nowadays, which is the amyloid fibril formation. We already talked a little bit in the beginning of the presentation, but this is what we follow. We have here the unfolding problem is that when you have a linear chain of residues or amino acids and they changes their structures in folds in a very, very specific way, in such a way that if the guy changes a little bit that structure, you may affect the protein function. The cell itself is able to recognize that change and say, wait, let's stop over there. This protein here is not continue to go to the cytosol or some organelle or whatever, is going to the digestion system of the cell to cleavate all the residues again, then those will be reused to make other proteins. Our system is able to recognize that. Since we have a lot of cell multiplications in the cell, if you don't have a very, very good protein control system, using the chaperones, using whatever, you will have some problems. The main problem is when our system is not able to identify that specific structure, or it's able to identify, but it's not able to cleavate their structures again. And this is the problem of when we have the amyloid fibril formation. So here is what we call the Fun theory for protein function. This is quite a physical representation. So in this X here, I have energy and I have entropy also. When the protein is in the native state, which is the protein was produced to be here. Here the protein is native. Any place else, it will not be in the native conformation and most likely will not be active at all. So here we have a very, very high energy, very, very high entropy because we have several possible conformations and this gives rise to this large entropy. As the protein is folding, it's losing the energy, it's decreasing also the freedom degrees, so you are decreasing also the entropy until it's this point here. However, by some reasons, by some places or by some misfunctioning somewhere, the protein can reach in those places here. When it forms amorphous aggregates, it starts to aggregate. Some protein that could cover some specific hydrophobic site was not able to work, I don't know. By some reason, you have that stuff over there. Then you attach, you may have this kind of amorphous aggregation. This kind of structure generally, depending on the case, of course, our body, our cell machinery is able to take a look and say, okay, this is not right. So it stops over here and go get this structure, put inside the digestion of the cell, cleavate everything, and you try to reuse what you can from there. Up to here, we are okay. This is happening all the time, tons of time, millions of times in our body. There is no problem at all. However, there is a specific position here that the protein falls into this very, very, very small energy, small entropy conformation, which means a very, very stable conformation. Remember, stability from the physical point of view is to go away from this guy here, come to the other side, for example. This is what we call stability. So as difficult as getting out of your place here, which means that smaller your energy is, and difficult is to break that energy because I don't have enough energy to give to you. To have an idea, it's very, very common to unfold proteins using SDS, sodium indoleucyl sulfate. But if you get those guys here and use SDS, you are not able to break those cross-beta structures. Those are very, very, very stable structures. And this is why I said from you in that first part that from the physical point of view, the diseases are very, very alike because all of them, the machinery is the same. What is changing is the protein or proteins, generally we have dozens of proteins involved in this process to make this amyloid fiber. The process is the same. The physical process is the same, but of course they are different from the point of view. So what you have from the schematic point of view, we have the monomer, we have a starting this cross-beta formation, then you have the protofibers, the amyloid fiber, and you have this entire machinery. What we know nowadays is that these are the toxic species for the cell, and those are toxic, those kill the cell. You have also a seeding effect. When the cell, by some reason, send those guys away, those are able to infect other cells and induce the proteins from the other cell to go with them in that structure to form the amyloid fibers. So what we are doing in that work, this is a collaboration with some guys in Argentina. It was a very, very nice study regarding Parkinson. Parkinson's a very, very, I think nowadays it's unnecessary to talk about the importance of studying Parkinson. It's a very, very huge problem that we are facing off because we are getting older, and we are having more cases of those kinds of diseases. The main point of Parkinson is that there is some specific region in the middle of the brain that will have your protein, everything is working fine, but when they start to aggregate, you may develop Parkinson. There are several different kinds of Parkinsons. And inside those aggregates, the main protein is alpha-synuclein. And there is also another very important protein over there, which is GAPDH. It's involved in the glycolytic cycle of the cell. And these are what we are seeing when we do electron microscopy. Those are the fibers. Those are oligomers. Those are the monomeric way. We know also that we have this kind of infection and induction. So we have the seeding problem. If you put a protofiber over there, you will induce the amyloid fibril formation of the other healthy proteins. So we must also have that in mind. These are some diseases that have the same mechanism of the amyloid fibril formation. This is quite old. I mean, it's 4 or 5 years old. So most likely we have different diseases over there. Just to call your attention, Parkinson's disease, Huntington's disease, diabetes mellitus type 2 disease, Alzheimer's disease, all of them. Oh, this is the crazy call disease, this number 5 here. All of them has the same way of working, which is the one I explained to you. The main difference is the protein related over there. So in order to tackle that problem, we used small angle X-ray scattering to follow the early stages of the amyloid fibril formation of this specific protein, GAPDH, in the presence of heparin. Because we also find inside the patients that died with Parkinson, the presence of high amounts of this kind of polysaccharides, polymers in the presence of the fibers. So just to go directly to the results, here I have the protein scattering a long time. In that model, the protein is a homotetramer. So we suppose that we have, for example, the protein tetramer, the dimer, and the cylinder. And we try to find how much was their contribution a long time. And we see that the tetramer concentration is decreasing. The dimer concentration is more or less constant, and the cylinder concentration is increasing. This is a is a good information, or a good first point, that we have our homotetramer splitting into dimers, and those dimers are somehow aggregating in solution. So our collaborators, they make molecular dynamic simulations, use those proteins to try to see how could be those dimers conformations inside this structure. This is the structure that they get from the molecular dynamic simulations, and this is the P of R of this guy here from the molecular dynamic simulations, and the P of R of this cylinder that I use. And remember that now, when I'm talking about the cylinder, we come back to the beginning of the class when I showed to you the geometric models. We used spheres, we used ellipsoids. In that case, we use just regular cylinders. So even using regular cylinders, we were able to describe quite nicely this structure that was confirmed with using molecular dynamic simulations. This is only to show to you a strata. We have this trimer of dimers. We have this interaction spot here in the middle. This is only to see that this is the same monomer in the different layers of the protein, and you have this kind of twist. And this kind of twist is very, very common from amyloid-like structures in solution. And confirm, I mean, being another information that this most likely is the structure that I have on my aggregate. Okay, now I am asking to you, do you guys have some doubt regarding these examples? Do you like that I can explain something better or different, or you are okay? Yeah.
Mm-hmm. You can do it exactly in the Fourier space, and later you go back for the real space, because the summation in real space and Fourier space, it's equivalent. You can change this.
Yeah, they are equivalent. The main problem is that we have not only a space problem, but a dimension problem. We have in the Fourier space a one-dimension information. The I of Q against Q. I have just one. And when I'm going to the real space, I have the balls in 3 dimensions, X, Y, and Z positions. So besides that, we have to take this in account, that you have a difference not only in that, but we have only when you have this kind of stuff. In the 70s and 80s, it was very, very common when the guys would like to, it's not so common, but I mean, it was the state of the art in that period, when you are studying the electron map density of different scattering particles or systems like cubic structures of lipids, you have the electron density map, you have a 2D information, and then you try to, you have, okay, what we call the phase problem, but then you may try to get this information using these 2D maps. But even though you have what we call the phase problem, because we have the problem of X-ray, that if I have a scattering particle in the vacuum, or if I have everything full of matter and the whole, I have the same scattering. If I have the matter as compared to nothing, and nothing compared to matter, I have the same scattering if they have the same geometry. So we have this, the Babinets principle in that we have to also take into account. But there are several different approaches that you may use to try to understand your problem. The problem is that we make the measurement in the reciprocal space, and we would like to get information in the real world. To do that, you must do your Fourier transformation, and it's not unique, the solution. I mean, you may have different 3D conformations giving rise to the same or very similar experimental or theoretical, but able to fit your experimental data. This is our problem. It's not unique. So in order to try to avoid it, we try to repeat this procedure several times. There are several software that can help us in doing that. There are software of clusterizations also that try to get this whole bunch of dozen of structures and try to see, oh, this one is similar to that, but not to that, and this, to try to see how many populations you may have. But this is a very, very difficult problem to access. Not so easy, not so trivial to access that problem. Yeah.
Would you mind to explain a little bit more about the model that you use in SACS to get the measurement, the gradient, or the diameter?
Here, in this last example. Okay, let's see. For example, here, I don't know, the main problem, so when we are making the measurement, we have this scattering curve over here. we have this procedure that I was talking to him that try to go. We are in the reciprocal space because we make the measurements in the cool space. We can try to go to the real space. We can try to get some information in the real space to try to solve our problem. Of course, there are several approximations that I'm not talking to you. I'm putting away in this explanation that you must take into account, like monodispersed of your system. Your system must be what we call well-behavior, because otherwise you are not possible to go to make this transition from the from the reciprocal space to the real space. When we have, for example, this P of R, then just to look into the P of R, it's possible to get some general information. For example, in that case here, remember, I don't know nothing about the protein structure yet. But I know that I have, for example, the maximum dimension is 140 angstroms. This is the maximum dimension of the protein. I mean, I will not find two pieces of protein far away from each other, larger than 140 or something like this. So this is the maximum dimension of my protein. Then, do you know we have a kind of inflection point somewhere here? This inflection point is on the order of magnitude, more or less, of the cylinder cross-section. So I have an elongated structure, and this cross-section is roughly, very roughly, something around 25, 30 angstroms. And this is just looking for that. So I have an elongated structure with a cross-section of 35 angstroms, or 25 angstroms, or 30 angstroms, or something similar to that. This is what we can do talking about the P of R. Up to here, I mean, you don't have so much complaint regarding your analysis. The problem is when we make these further steps. For example, in order to go from here to here, there is a lot of issues, Because when you use the Fourier transform, it's an integral that you must do in all the space. Even if you go to the real space to the reciprocal one, or to the reciprocal one to the real one. The problem is that I don't have the information of the entire reciprocal space, because I have a first experimental point, and the last experimental point. From 0 to the first one, I don't have any information regarding this. And from the maximum, far away from that, for larger values, I don't have any information from that. Those information we throw away. This information we get from the Guinea law. And there are several people around the world that doesn't agree with this kind of analysis, that it's very loose. And it is. But it's enough for us to get the information that we need to give just the general structural features of these proteins. For example, after then we propose these positions. This is drawing. I may have some different changes here or different changes here. This is a proposition. We are proposing that inside that elongated structure, using all the information that we got, using this crystallographic structure from other proteins from different animals or organisms, we try to think that and we believe that dynamics. This is what we can do with SACS, because it's a small resolution technique. We don't have a large resolution. I mean, large is smaller values. No, I mean, 20 angstroms, 30 angstroms, and this is what we can do using small angle scattering. Sometimes, depending on your problem, this is fine for you. Depending on what you would like to answer, it's fine. But in other cases, it's not. You must go to high-resolution techniques, like nowadays cryo-EM for larger proteins, or NMR for small proteins. Those are able to give you crystallography as well, to give you better information as compared to SACS. The advantage of SACS is the data analysis, which is not so hard as compared to them. Okay? Yeah.
Will be using this technique in membrane proteins?
To use for membrane proteins? Yeah. There are several people using small angle and small neutron scattering to study membrane proteins, not only for the structure but from the dynamics also. Since let's say the 70s, for example, there is a professor. He is in the ILL, Instituto Lau Langevin at Grenoble, Professor Giuseppe Zaccai. He's very known in the scattering area, and working with neutrons, and neutrons is fine because you have the elastic and inelastic contributions. Then you also take some dynamic information of your system. He studied, for example, bacterial rhodopsin, the proton pump of the bacterial rhodopsin in membranes. So it's possible to study. I myself, during my PhD, during my postdoc, we made some experiments using this information. And the problem in that case is that you have a mixture of, you have the signal from the protein, the signal from the membrane, and the cross term. The electron that is inside the proton with the electron that is near the lipid, which is not so easy to deal with. It's not an easy task. It's possible, but it's not easy. Tomorrow, I will talk to you about nanoparticles, more colloidal systems. We are not in talk more about proteins. I left the day today of proteins. Tomorrow, I will show you another examples for using drug delivery systems and stuff more on that regard as compared to proteins. But even though if I have some question, I will be here today, the entire day. Tomorrow also, Friday, come back, I will be here with you. And I think I was done. I was just to remember you. But of course, you take note about the meetings that we are taking place here in Brazil. Those will be very, very nice for researchers all over around. Of course, I showed to you this measurement and scattering curves, but most of them were not performed by me, but for the students. All of them are very, very nice and hard worker. Any one of them is working with a quite slightly system, one more similar to the others, some others not. And this is our group. If you have some questions, please feel free to talk to us. And that's it, and thank you very much for your attention. Some other question? Can we go to lunch? Thank you.